Hello. Today I have the pleasure of being joined by Dr. Aaron Sato, who has over 25 years of experience in biotechnology, focusing on discovering and developing novel first-in-class antibody therapeutics. Aaron was recently recruited to Adimab several months ago as their new Chief Strategy Officer. Aaron, we are really delighted to have you here today. You've had a number of roles in large pharma biotech as well as startups over the years, developing antibody-based therapeutics. Tell us about your new role at Adimab and what drew you to this opportunity.
Thanks, Jennifer. I'd love to talk about it. So over the last 25 years, my career in both biotechnology and drug discovery companies often involved advising collaborators and internal teams on advancing therapeutics to the clinic. So this consultative experience really led me to think about a kind of new career for myself. And it really got me thinking about a new type of role. And it got me thinking that maybe a chief strategy officer might be a great next career step for me. And so right around the same time, my colleague back from my DIAX days, which was actually my very first job in biotech, Guy Van Meter, who's actually the chief business officer at Adimab, called me up and he was actually looking for this exact same role. So it was actually really great timing for me. And so I actually saw this as an ideal opportunity to actually join one of the most established, well-known, antibody discovery companies in the industry to actually work alongside some of the most talented protein engineers in the business. So in my current role at Adimab, I oversee marketing strategies and partner engagements across various therapeutic areas, including antibody discovery, T-cell engagers, and multi-specifics and TCRs. I focus on competitive intelligence and technology analysis and building relations across helping drug discovery companies and industry leaders while working closely with business development to really help them grow the business at Adimab.
Thanks for that answer. It sounds like the stars really aligned and converged for all parties for this new position. So congratulations to you on this role.
Yeah, super
And
excited.
along those lines, what are your current thoughts on the state of antibody discovery, engineering, and development?
Yeah, great question. So in terms of antibody discovery, I mean, I believe that currently, it's really split between kind of in vivo approaches and in vitro approaches right now. But as you probably realize, in vivo and silico kind of de novo discovery is really, coming up as kind of like that third pillar of discovery approaches. Is it ready for prime time right now? In my opinion, not quite yet, but I think in the next couple of years, it is definitely going to be ready for prime time. And I think at that time, it's really going to kind of usher in a new era of drug discovery terms of engineering, I think, in my opinion, in vitro approaches still are kind of the main approach for engineering. And certainly with the advances in a lot of DNA technology, it's really allowing us to really make really sophisticated antibody libraries that we can then use, either yeast display or phage display. And currently, and as since I work now at Adimab, we have an amazing platform for using these libraries to really select for really defined specificities using our platform. For example, we can select for very pH-dependent binders using our FACS, using FACS to actually pull out those specific binders. And this again can be, and I'm really excited too about using a lot of these in silico approaches in the future. For example, machine learning approaches to even further optimize candidates as well. And then finally, on the development side, of course, COVID-19 really helped us kind of speed up the process of developing antibodies. And certainly, the government and the FDA are actually putting in place a lot of regulations to actually learn from a lot of those development that we did back in COVID-19. And even in the last year, they're putting a lot of regulations in place to allow us to kind of use platform processes from that to actually speed up the process of antibody development.
Sounds like a lot of really exciting technology advancements that are in the pipeline and going to be coming forth in the future. I'm wondering, do you think that all the low-hanging fruit has been picked in terms of disease targets and indications?
Yeah, great question. I mean, in my opinion, while I think the traditional validated single antibody targets, really represent the first wave of therapeutic development, and those again were the kind of the low-hanging fruit. in my opinion, really the next wave of kind of low-hanging fruit is really the combination of those low-hanging targets. And because now we can, basically create higher order bispecifics and multispecifics using, even now in Adimab, we have these amazing technologies for creating those higher order bi and multispecifics. We can then create those combinations of antibodies together to create those more sophisticated biologics. So I think now the lower hanging fruit are those types of molecules. And so we just need to be more sophisticated and smarter as we create those combinations. And again, we'll have to have a lot of data in terms of those target combinations to then create those higher order bi and multi-specifics. But I think there's still a lot of creative ways to create those combinations of targets in the future.
So combination targets, bispecifics, you mentioned AI and machine learning a little bit. What technology innovations do you think are most critical to disruption and advancement in the field?
Yeah, to your point, yeah, again, I'm really excited about kind of AI and its silica-based approaches moving forward in the future. One area that I'm super excited about is how you can use AI and machine learning to optimize candidates. One area that I've done a lot of work with in the past are kind of zero shot and few shot models for doing antibody optimization. Again, this allows you to, do either like really fast mutations in antibodies for either increased affinity or increased stability. I think this is again, like I said, is just giving you kind of modest increases in optimization currently, but I think in the future, we could really see dramatic increases in stability and affinity with antibodies. The other area that I think is another big area that I'm really excited about is using NGS sequencing of either immune repertoires or repertoires of our panning that we're doing without a yeast display or in vitro selections to really allow us to pull out even more sequences from our in vitro panning or our immune repertoires as we're doing immunization. I think that's another way to get even more value from our antibody discovery work. And then finally, having worked at another company that was a DNA company, I'm also really excited how we can use a lot of the advancements in DNA synthesis and in SynBio in particular to enable us to make even bigger and more sophisticated antibody libraries. And we're going to continue to learn from that field as they have more and more advancements. We're going to be able to make better and better and better naive libraries as well as optimization libraries as that field advances as well.
Great answers to the question. Really looking forward to you and others realizing those visions. And I guess final question that I have is we'd love to know more about your general vision for the future. If you look 5 to 10 years ahead, what would you have liked to accomplish in your new role?
Yeah, that's great. So looking ahead to my role 5 to 10 years in the future, I would have really loved to, basically kind of check off four key milestones in my role. First of all, I'd really love to establish Adimab as being recognized in life sciences as being the predominant player in antibody discovery and optimization, as well as all the other technologies that they've been pushing for. Again, they already have a fantastic brand name in this field, and I want to just again continue to propagate that brand throughout the industry, and really hope to kind of propagate that through all of the digital channels that we have available with us today. Second, as we were talking about before, AI and in silico approaches are really starting to be the third wave of antibody discovery and optimization. And I think Adimab is poised to really play a major role in that with our amazing platform for screening antibodies, as well as, we've been around for 17 years. We actually have a lot of data as well as a huge expertise in antibody developability. So I think we're poised to actually play a major role in this field as well. And I want us to actually step into this as well. So we're planning to really play a major role in this third wave as well. And then thirdly, I also want us to see if we can step into other markets as well. And so we're trying to see if we can step into like other, for example, Asian markets. Could we get more involved in China, Japan, Korea, other Asian markets to see if we could establish more partnerships in these markets. And then finally, continue to continuously evolve our technology. We always need to stay current. And the awesome thing about Adimab is we continuously evolve our technology. We always continue to reinvest in our platform. We actually spend a considerable amount of our budget reinvesting in our platform. We always want to stay current. And so and I want to actually help and contribute to that by actually contributing ideas to the company. And so I hope that through my role, I can actually help to actually contribute to that continuous evolution and process.
Thank you so much for sharing your thoughts and insights with us today, Aaron. We really appreciate it. And we look forward to seeing you and everyone else at the PEGS meeting in May.